Hello guys, you are watching Dr. Varun on chlorophyll classes. Well, for my 12th student, this video is for you. We were studying what? We were studying at the principle of inheritance and variations, that is from the genetics, and we're gonna be continuing with it. In the last class, if you remember, I gave you certain questions. Okay, what were those questions? Let me just check up. Those questions were regarding the what would be the possible genotype of genotype of the gametes. Okay, so these were the two questions which I gave to you. That is, here you have four different types of like that: X, Y, Z and A. These types of heterozygous genes available. So over here only I am making up space for it. So the answer for this one would be what? Then we will go further with it. Okay, so the question was XX, YY, ZZ, AA. So now how many gametes would be possible? I told you that to find the number of gametes that is possible by organisms with a particular type of genes, then it would be what? 2 raised to power n, where n is the number of heterozygous genes. Okay, so that is 2 into 1 2 3 4, that is 4. That is 2 into 2 into 2 into 2, that is 2 to the 4, 4 to the 8, 8 to the 16. So 16 different types of gametes would be there. Now if you have to make it, how you will do it? I told you that always start with what? Always start with your initial one by one. So capital X, small, then each of them will have the YY. So capital Y, small y, capital Y, small y. Move further, then it has capital small, capital small, capital small, capital small, and the last one would be what? Capital A and small a with everyone. So this becomes what? This becomes total 16 such. Okay, all are different ones. I hope this thing is understood. Another question that I have given to you, that is what? It was capital A but capital small a, then both the this are what? Same. Okay, I will just copy and over here. Okay, this thing, this is the second question. So here I have told you that the number of gametes would be told to you with the help of what? With the help of a formula, that is what? 2 raised to power n. How many different types of heterozygous n would be? N value would be what? So this is the n value 1, this is same, and this, so means that is 2, that is 2 raised to power 2, that is 2 into 2, that is 4 types of gametes only will form. Let's just check it out. If we have to make it, go through this, very important, because this would be the question for your NEET or your board exams, not the other ones. Easy one things never comes, these things will come. Okay, so what it is A and A, two options, both. Okay, both will has what? Wait, with respect to B, they have what? Same thing only, no difference, because capital B only. So I will be writing it what? Only option this, one option only. Okay, either you take one capital B or other capital B, it will be capital B only. Then for C also they have only one option, that is this small c. But now the options for the D would be two. What are they? Capital D, small d, capital D, small d. With respect to E, again the option is just one, so they will have this. So these are the 4 types of gamete for this one. I hope this thing is understood to you. If not, then let me know in the comment section. I will make a short video for it and share it with you. Okay, moving on to the today's topic. That is what? That okay, these things has been seen in the genetics, but that what are, like, it will always follow the trend? This is the major questions. So now, once the Mendel did this, he published his work, but his work do not get the appreciation. We will learn it about it. But for now, I want to tell you that, think about it, what Mendel did, why his work is not appreciated. The reason that his work is not appreciated, beta, because his work was out of the league. Think about you also when you come to know that anything, okay, which is not known, he talking about factors, certain things which is inherited from parents to the next offspring, it is not very acceptable to the masses, okay, what he have done. And at that time, the worlds and the scientists and the discoverers were talking and busy about thinking, discussing about the Darwin theory of origin. Although the Darwin theory has a problem in itself, we will learn later on in the evolution topic when it will come. Okay, so over there also this thing will link. For now, the one problem was this, that was the era of Darwin. No one was really interested into what Mendel has done, the out of the league thing. Moving on to the next thing, that is he uses maths in it. Of course, we're going to be solving certain questions. Most probably tomorrow I will keep up more of the questions and we will solve it and we understand more thing about it. So what will happen, that for today's, the limitation would be that utilization of maths, taking the concepts of ratio, probability into the biology was out of the trend at that time and no one was accepting it. So the two limitations that we're going to be learning, and moving on to the Mendel's, when later what else then Mendel hasn't seen, because the things that Mendel encountered was not always true. Okay, so like this thing we have to see. So one by one we will be trying to learn few of it. Okay, so first thing today we're going to be learning the limitations or demerits of the Mendel's experiment. Limitation of Mendel, not just limitation, that would be different. Wait, limitation we will be learning later on, but first we will be writing what we have discussed, that why it was not acceptable. Okay, so question that could be asked to you is that why Mendel work isn't recognized or appreciated. Now we say that he's a founder of it, but at his time, why it is not being appreciated? So the answer to this question lies in that first thing, that Mendel utilize mathematics to explain the concept of biology, which was means earlier it was think that math has to do nothing with the living organisms. Okay, but he proved that no, math is there everywhere. So beta, math is everywhere. So those students who ran away from the math and said that only physics and little bit chemistry has math, in 12th beta, bio also has math. Okay, although the level of math is very less. Okay, just you have to know about 6th, 7th class math, but the math is there. Okay, so to explain the concept of biology, which was unacceptable to the scientific community, like he said that this a plus b whole square, that is a square plus 2ab plus b square, he said that the amount of expression would be in this form. Okay, means what? He said that if we are taking a monohybrid cross, then one A is representing one allele of the genes. You know about allele and genes. If no, then go back to see the first video and understand it, and then proceed forward to this video. Okay, that t plus t whole square, okay, means if we are taking a plant which is this, and we are making a selfing of it, then what we will get, we can find it by this equation. He is saying that. Okay, so that would be what he's saying, that t square plus 2t plus this, means in short he's saying that the ratio with the genes would be like this. Look over here. Normally what we used to do when a F, like this is what F2 hybrid now, okay, means F2 generation that will forming now. So in F2 generation, what would be the genotype? The F2 generation, the genotype is 1:2:1. This is only what is being shown over here. This means what? 1. This is what? 2. This is what? 1. Hence, this equation is in itself is working over here. Okay, so this was unacceptable at that time, but this has been explained to you. So in NCERT, they are explaining you this concept like this way. So you have to remember that this equation is like they are telling you to explain this thing in a bit more cumbersome way. Okay, I hope that this thing is understood. The first point. The second point that was problematic was what? It was an era of, or the period of Darwin, where Darwin's theory of origin of species was, everyone was busy and no one paid attention to the concepts of genetics, which was the base of the origin of species, which Darwin also don't know. Okay, we will study about it later. Okay, so it was era of Darwin, where scientific community was busy in discussing Darwin theory of evolution. Which book he wrote it, we learn about it later, but the concept was what? Origin of species. Origin of species book, where he explained about it, what he has witnessed and seen. Okay, so these were the two problems that were there. Now let's just see that. Okay, like Mendel died. Okay, no one paid attention to his work. Okay, now what happened after that? More research work keep on happening. Okay, what were the more researchers? That was post Mendel genetics. Mendel genetics was correct. Later on someone find out, we will study about it later in the different topic. For now, let's see that, okay, Mendel said something, we found it, that is correct also. But what was the problem? The problem was the laws he gave. It was not applicable all the time. Means these laws are not universal. Okay, so now one by one we will come up as a different scientist. Means one would be Priyanka, one would be Nitish, whosoever is on my community. Okay, on this small community of, by this video, 181 subscribers. Let's say that one is Priyanka, one is Nitish, one is another student over here, and you just let me know your names also in the comment sections, because then it would be much easier for me to remember, at least for the time being, who was the initial ones. Okay, so if there are students from my old where I was working, if you were there, I know about the Nitish, but rest about, I don't know whether you are watching my videos or not. So if it is there, just let me know in the comment section, right? So coming back to the basics, that is what happened. Now we're going to be doing what? Each of us will one by one come and we will say that Mendel, you are wrong. Okay, because now we are the researchers and we have to see that whether he's right or not. Okay, so that is what this topic is, post Mendel genetics. Okay, so we're gonna be going beyond Mendel and we're gonna be challenging the Mendel's law of inheritance, and one by one we will shatter it. Okay, so post Mendel or Mendelian genetics. Okay, now first one that we're gonna be see is that, okay, the Nitish is going and exploring the things. Okay, imagine that I am that person. I'm exploring and I saw a plant. It was snapdragon plant, mirabilis plant, 4 o'clock plant. Other names are there. This 4 o'clock name, it means it is a vernacular name. Okay, so remember this thing. Or snapdragon, it is what? Vernacular name. Okay, mirabilis is the, or the anti-renus snapdragon. These are the names, okay, we're gonna be using up. So he saw a plant. Okay, it was what? Snapdragon plant. In the Snapdragon plant, when he's looking, he saw that I want to do experiment on this. Which experiment he want to do? He want to see that whether the law of Mendel's are applicable to this one or not. Which law? He want to see the law of dominance. Tell me what law of dominance is. Recall it. It says that the F1 hybrid, the contrasting character which will show its effect or express phenotypically would be the dominant one, and the other would be the recessive one, because no one changed itself. Let's just see that whether it happens with this plant or not. We took the plants. There are two contrasting characters with respect to its flower, a red color flower or a white color flower. So we assume that a red color flower is there, white color flower is there. I am saying that what will happen in it. Okay, so we are taking that. So we are studying about a concept called as incomplete dominance. This is called as what? Blending inheritance. Blending inheritance. Let's just do the experiment and then see that why we gave this name or something like this. Okay, so we took a red color flower plant. Imagine that this is the red color flower plant. Okay, and the same one has another variety also, that is what? White color flower. Okay, so now let's just write about it, basic things. So we have a red color and a white colored. Now we say that red colors are dominant one. So we are telling you what? It will show with the capitals. So it is a pure homozygous plant only we take. Okay, and this is small, we take it from small r. Okay, so this is one parent, this is the second parent. So this is what? Your parents. Okay, we have to just remember the concepts of genetics which I have told you in last two videos. If you haven't seen it, go see this first and come back. Okay, now what will happen? They will make gametes. If we have to do the sexual reproduction, they has to make the gametes. So they will make the gametes. What the gametes will look like? This will form only the R type of gametes. This will form with respect to small r types of gametes. So these are what? These are the gametes. Now these gametes, what will happen? Then there will be crossing. When we will do crossing, what will happen? Capital R, small r. This would be the first zygote that will form and the F1 generation. So this is F1 generation. Okay, now this F1 generation, if we see, this is a hybrid. Phenotype wise, how it should look? According to Mendel, it should have a look of what? The capital R. The capital R, I gave it to the red one, but that never happens. Here comes the magic. So what is the magic? The magic is that this appears to be pink color. Phenotype is pink color. So now the flower which came over here, it is what? Pink in color, like this. So the red and white, neither of it comes. Something comes in between of it. Because if you have played with the colors in your young age, what happened? If you mix a red color with a white color, the shade of pink will form. Means it becomes what? Incomplete. Not even complete red, not even complete white. That's why the name incomplete dominance. Plus, when you mix the color, means you are blending the color. When you're blending a red and a white one, you are getting what? A pink color. And this is what? Blending inheritance. The color has been blended together. We are not going on to the level of molecularity, how it is happening, which we will learn it later. Okay, but here we are trying to understand this, that something different occurs over here when I took the snapdragon plant. Okay, so now, okay, F1 generation this forms. Let's see that F2 generation what will form. We will do what? Crossing. Okay, till now what we know? We know that if both capital R will be there, then the red will form. If both small r will be there, then we'll form the white one. And if there will be hybrid, means capital R also, small r also there, then pink. Three different colors are there. Let's see the F2 generation, what is happening. In F2 generation, whether these colors are coming back or not is a question of our debate right now. So what will happen? There will be selfing. So selfing what means? It will make gametes. What would be the gametes? One capital R, one small r. These were the gametes. So one parent would be here, another parent would be here. We're going to be making the Punnett square. Okay. So see beta, we have what? One parent, another parent, capital R, small r. One parent on this side I have wrote it. Second parent I am writing on this side. That is capital R, small r. What will form? This first box, capital R, small r. Sorry, capital R, capital R. This is capital R, small r, capital R, small r, small r, small r. So if you see over here, what is happening? This capital R, this will represent what? It will represent a red flower. So we have what? A red color flower with phenotype, sorry, with genotype this. Then we have what? This flower will appear like what? This will appear pink, because it is what? This is a pink. Okay, this is also what? Pink. So there will be what? Pink color flower, and its genotype would be this. And the last one would be what? This one, that is what? R. So there will be small r, means white color phenotype plant. So if I have to write it, this is phenotypic ratio. This one is the genotypic ratio. So what is the red? How many red was present? Phenotypic 1. This capital R, how much was there? 1 only. Pink, how many? 2. This 2. This is 1. This is 1. Did you see here? The genotype and phenotype is much different than what we have seen in the monohybrid cross. In monohybrid cross, what was there? If you have to make a monohybrid cross, then there will be what? There will be red with respect to white. These two color will be coming according to Mendel. Okay, according to Mendel, if he hasn't gone the experiment, he would have said that one red will come, white will come, red is the dominant one. So the phenotype ratio according to Mendel would be 3:1 and genotypic ratio would be 1:2:1. But this never happened. What never happened, beta? This never happened. This happened. Means over here, when the incomplete dominance will be there, then phenotype and genotypic ratio will not matches with the monohybrid cross. It will be like this, 1:2:1. Understand it, feed up in your mind these things, and then move on to this. Okay, this is very important for you. So questions can be, understanding would be like this way. They would be putting you that Mendel said this in the board examination, and in your conceptual questions, assertion reason also in your NEET can come, because 2025 has changed everything. Understanding, deep dive knowledge is more important than anything else, and questioning why is very important. So you can question things. If you are not getting the answers in this video, in the comment section, next video, whenever I will making and continuing with the chapter, I will give you the answers for it, or I will even make the shorts out of it. Okay, so this was about the incomplete dominance in F2. But incomplete dominance, if you have to see it, you will be seeing the F1 generation only. Okay, but if you want to see that how it is continuing further, then you will go to the F2 generation to see the difference in the phenotype and the genotype. One question that most of the students confuses that in F1 generation, what is the phenotype and genotypic ratio? Beta, genotype 100% would be same only. So don't confuse here. The genotype it will be only this, only and only 100% this only, no other things will come up. Okay, this thing should be understood. Go through it. Notes will be shared to you on the Telegram, as well as on the link would be in my WhatsApp channel as well about all of these things. Okay, Telegram for sure you will get all the things, if not on the links available, so you can easily download the PDF on the Telegram channel. The link would be in the description. Moving on, after the incomplete dominance, the next one for our today is that is codominance. The word itself speaks a lot more. That is what is codominance? Codominance means together. Okay, so here what happened? Simple. Co means together. Dominance means dominant effect. Means we are taking the contrasting traits, but both are showing in the organisms. What is this? Let's see that. The example we're going to be taking for this would be, okay, one thing is there that I am not explaining the like statements. Okay, that I'm not writing over here what are the statement. I'm just giving you the answers directly for this one, because understanding is more important than the statements. Later on we will come up with really good notes, so you can just simply download it, or you can even purchase from us, okay, if you want. So codominance, what is happening? We're going to be seeing them. Example we're going to be taking is, let's just give you example one itself, that have you seen cow with two colors like white and brown? Maybe there are patches of white and brown. Maybe you have seen cats or dogs with the patches, equal patches of white, brown, or something, white, black, something, equal patches. That's an important one. Dog example is different, but we are going with a cattle, that is your cow. Now what happened in them? In them the codominance occurs. Means most probably one parent would be white in color, the other parent would be brown in color. Okay, we call it red in color. Okay, so these things are there. So if that is the thing, then what is happening with the child? Okay, of course that is a brown, but scientific community don't call it brown, they call it red. Okay, scientific community is weird, I must say that. Okay, so we are taking the example of what beta? Example is the coat color. Means the outer fur is called as coat of an organism. Okay, so coat color of cattle. So now we are taking what? Two different cattles. We are not just refining ourselves to the plants. We are going beyond to the, what? Animals also. So over here we have one parent, this. Okay, this is not looking like bull, but imagine that this is the bull. So this is what? This is the capital R, that is Red Bull, one parent. And another is what? R, female. Okay, looks like dog, but these are cattles. Okay, don't make fun of my drawings. Okay, that capital R, this is white cow. Okay, we are giving it names. Okay, if these are the parents and their coat color is so different, then their gametes would also be different. What would be the gamete here? Capital R, small r, this is small r. Now when these gametes will fuse together, when there will be mating, crossing will occur, fertilization, we can even say the fertilization word, then it will form what? Capital R, small r. Now, according to Mendel, we are discussing with respect to Mendel. Why I am saying this? Because these gonna be the question beta. This way we have to approach the thinking towards the questions, because this will be coming in paper. That's the level of questions in the paper. So according to Mendel, this should be what? It should be capital R should be the dominant character. Law of dominance should be there. Okay, so again we are thrashing the law of dominance only. In this chapter we are thrashing that thing only. So he said, according to that, there should be a red color. Okay, but that never happened. The color goes to Rome. Phenotype. Means there will be patches of white and red. So means the organism, like the calf that will form, means the baby that will form, it will have what? White color also, and this color also. So for example, I am giving the colors like this. Whereas white color is this rest of the part. Of course, when we see it, we look like that white color has certain patches of it, but it is equal things. So this becomes what? Rome. Genotype is this, but both are showing its effect. Means in F1 generation, no one is dominant. No blending occurs. Both shows its effect. So in codominance, what happened? Both contrasting trait shows its effect or phenotype. That's why we call it codominance. Now let's see that what will happen in F2 generation. You will also start making with me, because seeing is easy, making is tough. So when I am also trying, you should also try. Okay, or you should stop it and try it, and then come back and see that what is happening. Now when we do the selfing, there will be parents. So we are doing what? Selfing between two of the Rome parents. Okay, so I am making the Punnett square. Male, female, capital R, small r, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, small r, small r. Now, if we see that this would be red in color, this would be mix. These two will be the mix, and this progeny would be white. So, if we are taking the phenotype in F2 generation, after selfing, F2 generation will come beta. Okay. So phenotypic ratio. Phenotype is what? Red will also come. Rome will also come. And white will also come. The Rome one is showing what? Codominance. Okay. So this one showing codominance. Showing codominance. Phenotypically. These things are being shown phenotypically. Genotypically, things might be anything. Okay, so that is 1:2:1. What is genotypic ratio? Genotypic ratio, that is capital R, capital R, capital R, small r, small r, small r. 1:2:1. So this thing is there. I hope this thing is understood to you. Codominance. Okay, moving on to the next one. Okay. So you're like, sir, it's not stopping yet. No, it's not stopping yet. We're gonna be studying more about it. Now the next topic is multiple allelism. Okay. That is multiple allelism. Now the word itself is telling you about what? That multiple means many. Allelism means allele related factors or phenomena. So alleles are what? Alleles are that a gene has a contrasting characters, and those contrasting characters are called as alleles. Now multiple alleles means what? For example, normally we say that one factor or one gene has a, like, of course it's a diploid organism, so there will be two, maximum it could be two. But if we see in the population, there could be more than that. How I will say that? Multiple allelism phenomena operates in population, first of all. Operates or visible or appreciable or observable in population. What? Let's go to it. For example, you know what? Blood groups. Now inside the body, I have what? My blood group is A. Okay, I'm not talking about positive or negative thing. Let's just say the basic things. Okay. If you don't know how it is working, we can go back to the 11th class. But here it's like a revision of 11th class also with respect to ABO blood grouping. Okay, so I'm talking about example is in ABO blood grouping. So I tell that my blood group is what? My blood group is A. Your blood group would be B, or another person's blood group would be O. Now this is the contrasting characters. What are contrasting characters means? A, B and O. These are three different alleles of the contrasting characters. Either it could be A, or it could be B, or it could be O. How it is possible that, like, we know that inside my body two things can be there. Either it could be AB, AO, BO, or A, or that is the case, right? How come this O is also in the like someone blood group, like three different genes are there? ABO blood group means what? That there are what? Three alleles. This three allele is questionable. So you will say that how, like the question that would be framed to you, that could be like that in a diploid organism, only or maximum two alleles contrasting could be present. How does ABO blood grouping has three different alleles? That is A, B and O. Good question, na? That how it is happening over there. So now the answer lies is that these three, answer is what? These three alleles are not present in one individual, but observed in population. And the organisms which are able to show the Mendel effects or other things which we have studied till now, according to that inheritance pattern, they are showing its effect. Let's just see that what is happening. Now over here, this, you have to remember the next factors or the facts about it. Now A and B is dominant allele, means it will show its effects, whereas O is recessive allele. With this factor, what we have to understand? We have to understand that to O to show its effect, it has to be in the homozygous recessive condition. Means both the alleles should be there, O present in my body, then only it will show. If by chance one of my allele is O, other allele is A, it will show what? A is the dominant character. One question. Okay. So for O to show phenotype, means O blood group, it has to be in homozygous condition. So, and now the question is, what about A and B? If both came together, then codominance occurs. Okay, this is by some people have blood group what? AB. Okay, and if A and B allele came together, then both will show phenotypically, both will show phenotypically, and blood group will be AB. Understood this? Let's just see that what is happening other way around, and we will see all the possible genotype and phenotype in the blood grouping. Okay, so here I am writing blood group. This is blood group. This is genotype. What could be the genotype possible with it? Then there should be what is a possible phenotype if that will occur. Now inside their body antigen would be present. So this thing is from the 11th class. So those who didn't pay attention in the blood circulatory system, here they can understand. Now they have antigen present, which will show it effects and give its blood type. There will be antibodies present, means against it. Can like donate blood to and accept blood to. So these things we have to remember. Okay, let's just see that one by one. If the blood group is A, how it is possible to have A? So this is present on what? Antigens. These are antigens are what? Proteinaceous in nature. So we will putting that immunoglobulin. So A, if the first allele is also A and the second is also allele, means homozygous dominant condition. Okay, then the phenotype would be what? A. Okay, expression would be blood group A. If it is one is A, another one is O. This O can be written as like A and small I, recessive character. Then also because A is what? A dominant character. So it will show it effects over the O and mask the activity just like Mendel dominant. So here it is following what? Here it follows Mendel law. Okay. And the antigen that is present, what? If it is showing A effect, means antigen also would be A. Antibody would be opposite of it. Now O antibodies are not present. Okay, so it is what? Anti-B antibodies would be present. Now it can donate blood to what? Only A, because if it give it to anyone else, like if it gives to, sorry, it can gives to A and AB, because AB also has A, so it can accept it. And it can accept blood from A and O, because O do not have any antibodies, so it won't do any problem. Okay, so means it do not contains any antigen, so antigen is not there, so its antibodies are not present, so O can be accepted. Okay, then comes the second one. After that, B blood group. So we can have IB, IB, both are the homozygous condition, or IB, IO, which can be written as IBI. Here also it follows Mendel's law, which is B or B. Whatever be the condition, the phenotype would be B. Antigen is also present, B. Antibody would be the opposite, that is anti-A. It can donate to what? It can donate to itself, B. It can also donate to AB blood group, whereas it can accept blood from B and it can accept blood from O. Let's move on to the next one. Then we have what? We have O blood group. So it has to be O, means small I, or IO. This way we can write it. It also follows Mendel law. It follows Mendel law. Phenotype would be what? O. Antigen, no antigen is present. Antibodies are present for A and B. Okay, so anti-A and anti-B antibodies are present. It can donate blood to any of it. Okay, so it can donate blood to O, it can donate to A, B and AB, but it can accept it? No. Okay, so it can only accept with the O. And we came up to the last blood group, that is AB blood group. And here the question forms. So sir will like, sir, why are you telling it at the end when the question forms from here? Because I want you to remember the other factors and then come back over here. Okay, now here it is what? It has A antigen, sorry, A and B alleles. So the phenotype becomes AB. Antigens would be what? A and B. Both the antigens would be present here. Antibodies. No antibodies would be present because both the antigens are present. So antibodies won't be there for it. Then it can donate blood to what? It can donate blood to AB. Okay. Only to the AB. It can't donate anyone else. Whereas whether it can accept blood, it can accept blood from A, B, O and AB. All. So this becomes what? This becomes universal acceptor. This becomes universal donor. Okay. So these two factors, we did it. So this is what? This feature becomes what? Universal donor. This feature forms it what? Universal acceptor. This is multiple allelism. That multiple alleles, how it's working and all in this phenomena. Let's just come to what another special thing about AB. Now AB is showing its effect. Means if AB blood group is there, means individually it will show effects. So this is a feature of what? Codominance. So this is the feature of what? This is codominance. So the question would be like this, that in ABO blood grouping, justify that it shows Mendel laws also and it shows codominance. So here it is what? Codominance. Codominance shows or follows codominance. Will you remember this? This is an important word, multiple allelism. Next question that would be there that we will be learning, that would be what? How many possible phenotype is there? A, B, O, A, B. 4 possible phenotype. How many genotype is there? 1, 2, 3, 4, 5, 6. 6 genotype. So this becomes the next pointer for you. That is what? In ABO blood grouping, means multiple allelism factor, possible phenotype is how many? A, B, O, A, B. That is 4. Possible genotype. Possible genotype is what? 6. That is IA, means homozygous condition, heterozygous condition, again homozygous B condition, heterozygous B condition, then this and then this. So this thing is there which you have to remember. The question that would be asked to you, here I am writing it. Justify how ABO blood grouping shows or follows Mendel law of dominance as well as codominance. So when this question will come to you, what would be your answer? So in case of A and B, what is happening? If this condition is there, or if this condition is there, it will show what? A and B respectively as dominant character. Means you will express what? That in F1 generation only one is showing its effects. That is, in F1 generation only one allele. Okay, better one would be like this. Only one allele shows phenotypically or express phenotypically. So it is what? It is the law of dominance. Okay. In case of AB blood group, both shows its effect. Hence, what will happen? Codominance. So this is how it is showing the codominance effect. Well, for today's video, we're gonna be ending up here. We normally keep it a video up to 1 minute, okay, but sorry, 1 hour, but this time we are keeping a little lower, okay, because the other thing will take more than 1 hour and I don't want to stretch it up. So 3 things we studied for today. What was that? We studied about the incomplete dominance, we studied about codominance, and we studied about the multiple allelism. All of these are super important concepts to understand, and this is how we took a steps from 10th class to the 12th class kind of a genetics. Go through it, this topic. Tomorrow there's going to be another topic which is turning more topics from this post Mendel genetics, and this thing is available in NCERT, what we have discussed in today. But tomorrow's topic is beyond NCERT, but they are important because every coaching institute is teaching you those things. So we're gonna be doing it on YouTube together so that we have an edge similar like what the coaching institute have. I hope you like this video. If you like this, give it a like, share it with your friends or the 12th class students who gonna be doing the genetics, and we will make it fun for you. If you didn't understand anything, comment me down. I will make shorts out of it. These notes would be available on my Telegram channel. Its link would be in the description of the videos, and the link would be even available on the WhatsApp channel as well. Follow on those 2 platforms for the regular updates about the things. We are coming up with really good and cool stuffs in the future also. Tomorrow's for the 12th class. We're gonna be continuing with these things. Super important class tomorrow. Complete 1 hour class would be there tomorrow. More than that could be also possible. So don't forget to hit the subscribe and the bell icon so that you never miss the video from it. Well, that's all for this video. Thank you so much.